Hey everyone, this is Mr. Mott. What we're going to discuss today is how do you determine the pH of a water sample using indicator solutions. Indicator solutions are solutions that contain a chemical that changes color depending on what pH the water sample is. The indicators we're going to use are bromothymol blue, phenolphthalein, alizarin yellow, thymol blue, Congo red, and cresol red. Here we have a well plate that has pHs 1 to pH 13 uh, in the wells. 4 drops of each pH solution go in there and 1 drop of the indicator. We can see that bromothymol blue from about pH 1 to 5 turns yellow. pH 6 is more of a yellow-green, pH 7 is a green-blue, and then 8 to 13 is blue. So one nice thing about bromothymol blue is that it's very easy to tell right away just by adding a drop in there whether something's acidic, basic, or right around neutral. And again, less than 7 is going to be acidic, more than 7 is going to be basic, and right at 7 is neutral. Phenolphthalein also is very distinct from pH 1 to 8. There's no color change, so it's colorless. And then 9 is pink, and then 10 to 13 is sort of a deeper pink, sometimes called fuchsia. Thymol blue has a lot of nice pastel colors. One disadvantage of thymol blue sometimes is that sometimes the colors are a little bit faint. But it does show a nice variety, and so it is a good indicator to use. Alizarin yellow is really interesting. It has some nice yellows and oranges in there. What you'll also notice is that pH 1, pH 2, and pH 3, when you put alizarin yellow in them, actually form a yellow precipitate. So it's a yellow solid that forms, and that's different than what we see for 4, 5, 6, and 7, and 8, which is just a yellow solution. So that's important to notice that sometimes precipitate do form with indicators. We see some more precipitates with Congo red, in pH 1 and 2, and I think also 3 as well. And the different shades of purples going into oranges as well. Last, we have cresol red. I always think about with cresol red sort of Easter egg colors, nice and vibrant pastel colors. And putting this all together, what we can do is create what's called a pH indicator reference chart. So here's a list of all the colors that the indicators turn in pHs 1 to 13. And we're going to use this as a reference guide to try to narrow down the pH. So if I have a water sample and I want to try to figure out what the pH is, I like to start out with bromothymol blue. As before, we mentioned that when you have a sample that turns yellow, the pH is between 1 and 5, so it really narrows down what area of the chart that we should be looking at, and also what pH we should be looking at. My sample kind of turns sort of an in-between yellow-green or green-blue color. It's probably in the neutral, somewhere around 6 or 7 being neutral. And then for 8, if it's 8 to 13, it means our samples turn blue. So let's say that we take our water sample and we put bromothymol blue in it, and our sample turns yellow. That tells me that its pH is between 1 and 5. So it helps narrow it down, but not quite enough to be super specific. Remembering that the pH scale is based on the log scale, there's actually a 10,000 times difference between pH 1 and 5. So narrowing it down more than that is definitely something that we need to do. The next is we need to choose another indicator that we can use to narrow down the pH range. So let's think about phenolphthalein. Would this give us any more information? So by that, what we want to know is, can we narrow down the pH range? And with phenolphthalein, we couldn't, because we already know it's going to be colorless. So putting phenolphthalein in our water sample would not help. So another option would be to use thymol blue. So let's say that we took our water sample and we added thymol blue and it turned yellow. That would tell me the pH is between 4 and 5. So really narrowing down the pH range. Trying to narrow that down a little bit further, we would want to choose an indicator that in the 4 and 5 range that has some different colors to it. And our only option would be Congo red. So I would take my water sample, I'd put Congo red in it, and I would see which sample, which color matched the best. All right, and that'd be a good start to being able to figure out the pH of my sample. We'd still want to check with using pH reference samples of known pHs next to it, which we'll cover in another video. Let's say that we had, going back, what would be some other options? Let's say that we used bromothymol blue and it turned yellow again. And then we chose alizarin yellow. Alizarin yellow is a nice one to choose because you're either going to get a yellow solution in that range or you're going to get a yellow precipitate. So if we got a yellow precipitate, that tells us we're between 1 and 3. Now at this point, we have 3 good options to choose from which indicator to use next. So what we would want to do is, I think the best one would be thymol blue. Thymol blue has 3 distinct colors in the pH range of 1 to 3. Congo red only has 2 distinct colors, same thing with Cresol red. So what we would then do, we'd take our water sample, 4 drops of it, add a drop of thymol blue, and hopefully it matched one of those colors pretty well. If we were in the neutral range, and I want to clarify something a little bit, 6 is not neutral, 6 is slightly acidic, but for bromothymol blue, you get sort of these different shades of colors, and it's a little bit hard to tell sometimes. Is it more yellow-green? Is it green-blue? Maybe it's kind of an in-between, you're not quite sure. Then you would think that you're in the 6 to 7 range. Now, there's only one option that would give me more information in that range, and that's Cresol red. So Cresol red has 2 different colors between 6 and 7, so that's going to help me narrow it down. All right. If we were in the basic range and we're using bromothymol blue again to help us identify that, we'd have a lot of options to choose. And I wouldn't say one is necessarily better than the other, but let's say that we chose phenolphthalein. So phenolphthalein is nice because at pH 8 it's going to be colorless. pH 9 is sort of a light pink, and then after between 10 and 13 it's a darker pink. So that could help sort of narrow things down quite a bit. So let's say we had our sample turned sort of a pinkish color, but you really weren't sure about the intensity of it. We could then test it with our water sample with thymol blue. Another option would have been alizarin yellow. I think the only ones we wouldn't want to do would be Congo red and Cresol red because those 2 show the same colors. Okay. So if we tested our sample with thymol blue, hopefully our sample would match one of those colors. For added verification, we could also do alizarin yellow. So that's sort of how you would work through the process. What's nice but also tricky is that it's a process. You really have to think about what does each piece of information tell you, and how does it help you narrow down the pH. Thanks for watching.